Dear students, in my previous lecture I have discussed with you about the structure of viruses and their classification, and today I will continue with the viruses, mainly focusing on the replication strategies used by different types of viruses. Now to start with, viruses are obligate parasites as they lack various vital functions required to generate energy and a host of small molecules and macromolecules. Now for all these requirements, they need a suitable host. However, there is a great deal of specificity between the virus-host relationship, and most of the viruses even carry enzymes that are mandatorily required for their replication, which their hosts may not possess. Now, the aim of the virus is to replicate itself. So, to do that, the virus must attach to a host cell surface, make an entry, make new copies of its genome, and get the new copies out of the cell. Now, there are 7 steps that are followed in virus replication. These are attachment of the virus to a host cell surface, entry of the virus, transcription of the viral genes into mRNA, translation of the mRNA into viral proteins, viral genome replication, virion assembly, and virion egress, that is exit from the cell. So here is the picture that depicts all the 7 steps of replication followed by any virus. Now we must understand that all these 7 steps provide a useful template. However, this is a generalized template. Not all the viruses follow all the 7 steps, and these 7 steps do not occur sequentially always, and even some viruses follow additional steps. In the later stages of replication, several stages can occur concurrently, like transcription, translation, replication of viral genome, virion assembly, and virion egress. All can progress at the same time. Now let us have a look to the receptors and co-receptors that a virion recognize. A virion actually have various surface protein molecules with which it recognizes the surface molecules present on the host. Those are mainly glycoprotein or glycolipid in nature, and these are termed as receptors. Now this interaction of virion and receptor is highly specific, just like a key fitting in its lock. Some viruses can even bind to a second type of receptor called co-receptor, and these receptors are present in the host cell plasma membrane, and on the inner surface these are coated with proteins like clathrin or caveolin. In at least some cases, some viruses are there that are independent of clathrin or caveolin. After binding to the receptor, there is a conformational change which enables the virion surface proteins to bind to the second type of receptor called co-receptor. Receptors and co-receptors have multitude of functions like virus attachment sites for interacting with chemokines and other growth factors, and mediating cell-to-cell contact and adhesion. So here are the examples of cell receptors used by many viruses. For example, poliovirus use CD155 as the cell receptor. HIV-1 uses CD4 as the cell receptor. Now it has been demonstrated that various receptors and co-receptors have folded domains in the outermost part, just like the immunoglobulin molecules. In this picture, there are 3 examples, like the CD4 receptor for HIV, CD155 for poliovirus, and ICAM1 for various rhinovirus strains. All these receptors have immunoglobulin-like folds. Now on the virus part, virion also has multiple sites, and each site is made up of one or more proteins with which it binds to the receptor or co-receptor. So this table exemplifies the different virion attachment sites, that is the virion surface proteins, which interacts with the receptors and co-receptors. The virus attachment sites are present mainly on the capsid surface, either within depressions as in case of poliovirus, or within ridges as in case of foot and mouth disease virus, or it may be present on specialized structures like knobs on adenovirus and spikes on rotavirus. Some virion surface proteins of enveloped virus can bind strongly with RBCs of various species and can form them clumped together, a process known as heme agglutination. The proteins involved are called heme agglutinins, and the viruses which can heme agglutinate are influenza viruses and measles viruses. Now, we should know the principle behind this binding of virion surface proteins and the receptor, that is the forces that mediate this binding. Now, generally the forces that are involved in this binding are non-covalent interactions like ionic interactions, hydrogen bonds, van der Waals forces. No covalent bond actually exists, and the sugar moieties are mainly involved in the forces that bind the two. Initially, a virion is weakly bound to a receptor, that is, a virion can detach itself at any time instant, and when the binding continues, the opportunities are there for more surface proteins to bind to one or more receptors, and the binding ultimately becomes irreversible. Now let us have a look that after successful attachment to the host cell surface, how a virus gets an entry into the host cell. Now after binding to the host cell receptor successfully, animal viruses now try to get an entry into the host cell. This can be done at the cell surface, or this can be done after crossing the cell surface and plasma membrane. They have to cross another membrane, that is the endosomal membrane, and this is mediated by endocytosis. Now endocytosis is done either by clathrin protein or caveolin proteins, and most of the viruses hijack one or more of these mechanisms to get a successful entry into the host cell. Even there are viruses which are independent of clathrin or caveolin mediated endocytosis. Now here is the example of adenovirus. We can see that it uses clathrin mediated endocytosis, and SV40, that is simian virus 40, it uses caveolin mediated endocytosis. Now when the virus is engulfed within the endosome then fuses with other vesicles called lysosome, forming endolysosome. This lowers the pH to 4.8 to 5, and this acidification is very important for the fusion of the envelope with the vesicle membrane. There is another transport system, that is the transport of hydrogen ion across the plasma membrane, that also acidifies the virion environment. Now entry of naked animal viruses into cells. Some naked viruses can make an entry through the pore present in the plasma membrane, or they can use receptor mediated endocytosis. First there is reversible attachment, then there is irreversible attachment, and then they are engulfed within the endosome and they remain in the exterior environment. They are not yet in the cytoplasm, they remain within the endosome. So this picture depicts how a virus attaches itself to the host cell surface and makes a successful entry into the host cell, and this stage is before uncoating. Now entry of enveloped animal viruses into the cells. It is just like naked animal viruses. First the attachment is reversible. followed by irreversible attachment, and the envelope can fuse with two mechanisms, one with the plasma membrane or another with the endosomal membrane. So this picture depicts how an enveloped virus fuses either with the plasma membrane or with the endosomal membrane. Now fusion does not occur spontaneously, that is, the lipid bilayers do not fuse in a spontaneous manner. For this there are proteins which help in the fusion process. These are called fusion proteins. So here in this table there are examples of some fusion proteins that mediate the fusion of virion surface protein and the receptor or the endosomal membrane. Now in case of entry of enveloped animal virus, what are the role of fusion proteins? Now fusion proteins are made from larger proteins and they are synthesized after cleavage, and the cleaved product remain linked through non-covalent bond or disulfide bond. Now these are found as dimers or trimers. Each monomer has at least two hydrophobic sequences. One is transmembrane sequence, another is fusion sequence. The fusion sequence first lies hidden and must become exposed before fusion to take place. Conformational changes occur after virion binds to the receptor and the fusion sequence become exposed. So when the fusion sequence becomes exposed, the two membranes are pulled together and the fusion occurs. So there are therefore two categories of membrane fusion. One is pH independent, that is followed by herpes viruses, HIV viruses, and another is acid triggered fusion followed by influenza viruses, measles viruses, rhabdoviruses, etc. Once the fusion sequence is exposed, it can insert into the membrane and further conformational change in the fusion protein pulls the two membranes together, and this process causes release of energy of fusion protein and causes irreversible change in shape. So after successful entry, it is meaningless if the genome is not delivered into the proper location. So the next step is intracellular transport. So after attachment and entry, for intracellular transport, the viral genome must enter a proper location, that is either cytoplasm or nucleus, and for this they take the help of host microtubules. Now let us have a look for the intracellular transport of RNA viruses and DNA viruses. Now in case of RNA viruses, it has been seen that most of the RNA viruses replicate in the cytoplasm. So they do not need to get transported within the nucleus. Exceptions are there like influenza viruses, which require the host cell machinery from the nucleus, that is the enzymes present there are required for viral replication. So influenza viral genome must be transported within the nucleus. Now in case of retroviruses, it has been seen that the DNA copy of the RNA is done in the cytoplasm, and then they wait in the cytoplasm for mitosis to occur so that the genome gets delivered to the nucleus only in dividing cells. So the DNA to DNA copy is being made within the nucleus. As mitosis begins, the nuclear envelope is broken down and the DNA gets delivered within the nuclear compartment. Unlike retroviruses, in case of lentivirus, the genome gets delivered to the nucleus in intact form and in the intact nucleus, so it does not require dividing stage. This can occur in non-dividing cells. Now let us see the intracellular transport of DNA viruses. Most DNA viruses replicate in the nucleus, so their genome must be transferred first to the nuclear envelope and then across it in the nuclear compartment. Now for this, they get attached to the microtubules and then they are transferred to a location near the nucleus. The smaller nucleocapsids can enter directly through the channel at the center of the nuclear pore, and for larger viruses, the larger nucleocapsids, they must shed some of the structures to form slimmer structures so that they can get an entry to the nuclear compartment. So this is the picture which describes how the virion gets attached to the host microtubules and gets transferred to the location near the nucleus, that is the centrosome, so that it can make an entry through the nuclear pore inside the nucleus. Some DNA viruses like iridoviruses and pox viruses, however, replicate their genome in the cytoplasm. Now, after successful intracellular transport, that is getting delivered the genome to the proper location in the cell, then uncoating occurs, that is shedding of the capsid either partially or completely. This can occur at the cell surface, inside the cytoplasm, at the nuclear envelope or within the nucleus. Now, successful entry of a virion into a cell is not always followed by viral replication. The infectivity may be inactivated by intracellular defenses like lysosomal enzymes. In some cases, the virus may cause latent infection than a complete replication cycle. If, however, the uncoated viral genome remains intact at the correct location of the cell, then transcription and also translation can begin. Having described the attachment, entry, uncoating of animal viruses, now let us shift our attention to the bacteriophages. Now we know that bacteriophages are phages or viruses that infects bacteria. Like animal viruses, bacteriophages also bind to specific receptors and co-receptors, and the interactions are highly specific. For many phages, these molecules are on the surface of host cell wall or on other structures like pili, flagella or capsule. Many of the virus attachment sites are present on the viral tail fibers like the T-even phage, that is T4 phage. As with animal viruses, the attachment is first reversible and then ultimately the attachment becomes more stable and irreversible. Now a bit detail, taking the example of T4 phage, let us see how it attaches to the host cell surface and delivers its genome into the host cell. Now we can see the structure of a T4 phage with tail fibers, base plate with short pins extending from the base plate. Now the phage first attaches itself to the host cell receptor with the help of its tail fibers, and this is being accelerated by ATP hydrolysis, low pH, presence of magnesium and also presence of L-tryptophan. The base plate then settles down on the cell wall with the help of short pins extending from it. Now conformational changes occur in the base plate and tail sheath. The sheath becomes shorter and wider, and then the central tube is pushed along the cell wall and then across the plasma membrane with the help of protein activity of GP5 that creates a pore in the plasma membrane. Now ultimately the genome gets delivered from the phage head through the tail tube into the host cell cytoplasm. So this picture clearly depicts sequentially how it attaches itself successfully on the host cell surface and delivers its genome into the host cell. Now, interestingly, these bacteriophages follow two modes of replication. One is lytic, another is lysogenic. So to describe the Lytic cycle, let us take the example of this T4 phage only. What is that? Now Lytic cycle is followed by virulent phages or lytic phages. What is being done? The T4 phage first attaches itself to the host cell surface, delivers its genome, and the phage DNA circularizes once within the host cell and it is separated from the bacterial chromosome. The phage DNA then replicates and then those DNAs are assembled within phage heads. Then the progeny virions lyse the cell and gets out of the host cell. So this is the lytic mode of replication followed by most of the T-even phages. So this is another diagram depicting in detail the adsorption process and how it makes an entry and how it gets out of the cell after successful replication. So we can see here that the phage T4 first makes an adsorption on the host cell surface, then it penetrates and delivers the genome which circularizes. Then there occurs the synthesis of early proteins that are required for viral replication. Then occurs the replication of viral DNA, synthesis of late proteins, and the viral genome is packaged into the phage head and all the progeny variants lyse the cell and gets out of the host cell. So this is in totality the lytic mode of replication. Now to describe the lysogenic mode, the best example is the lambda phage. Now what is this lysogenic cycle? The phage infects the host cell just like lytic phage, but unlike the lytic cycle, here the phage genome gets integrated with the bacterial chromosome and it replicates as a part of the host genome, and that is also transferred to the progeny cells. So the lysogen and the prophage remains as a genetic symbiont, stable genetic symbiont, as long as the host is unaffected. But in case of adverse conditions, this phage DNA gets excised out of the bacterial chromosome and switches over to the lytic mode, and then again it replicates its genome, gets assembled into the phage head and gets out of the cell just like the lytic mode. So there are two proteins which actually regulates which cycle to choose, whether lysogenic or lytic. So the two important proteins that are involved in this decision making process, that is the C1 protein and CRO protein. Now we can see that when the bacteria isn't growing very well, then the lytic phase is inhibited. That is, the C1 protein activates the C1 promoter and inactivates the CRO promoter. And if the C1 protein gets activated, then lysogenic phase will progress. But whenever the host is in adverse condition, the C1 protein accumulation will be inhibited by the CRO protein, as because CRO protein activates the CRO promoter and inactivates the C1 promoter, then the lytic phase will progress. So the decision making of lysogenic or lytic cycle depends on the condition of the host. A temperate phage can integrate at specific site on bacterial chromosome and maintains a stable transmissible association and replicates along with the host genome. So lysogen and vegetative growths are just alternates. As long as the host cell is intact, an integrated prophage can survive as a stable genetic symbiont, but whenever there is any damage to the cell, the phage DNA gets excised out of the bacterial chromosome and it replicates and switches over to the lytic mode. So thus, lysogeny in bacteriophages is determined by the survival value that it provides to the phage. Now lastly, we have described the animal viruses and bacteriophages, their replication mode. Now let's have a short look into the plant viruses. Now plant viruses enter the cytoplasm of plant cells through a physical break that is done by mechanical damage or animal vectors. Once introduced, uncoating takes place in the cytoplasm and the released genome can move around the plant cell through plasmodesmata. So here is a very good example, that is TMV virus, tobacco mosaic virus, causing the tobacco mosaic disease of obviously the tobacco plant. So this is the structure of TMV virus, which shows in enlarged form the arrangement of the capsomeres, that is the capsid, and the RNA is arranged spirally within the capsomere. The capsomere here is also spiral. Now lastly, let's see the life cycle of TMV virus, that is the replication cycle. It starts with infection of the virus within the host cell, degrading the viral protein sheath, and then as the genome is RNA, first complementary RNA is made, and from that complementary RNA, newly synthesized viral RNAs are produced. After synthesis of newly synthesized viral RNAs from complementary RNA, those RNAs gets arranged within the capsomeres and full progeny variants are produced, and this completes the life cycle of TMV virus. So that's the end, and hope you have understood all the nitty-gritties of viral replication and how the virus overpowers the host cell machinery to replicate into progeny variants. Thank you.